the path-breaking research done by our resource person for today's program, Shri Mukesh Sharma, along with his resource guide, Professor Mihir Kumar Purkit from the Department of Chemical Engineering of Indian Institute of Technology, Guwahati, has been extensively covered by different national as well as regional print media. Let us see what the media tells us about Shri Sharma's new invention.
Shri Mukesh Sharma's new device, which can turn wastewater into energy, is also covered by different electronic media. India Science TV channel, a 24/7 satellite channel dedicated to science and technology under the Department of Science and Technology of Government of India, also covered it beautifully in its Science Monitor program. Let us watch what India Science channel thinks about the research.
Researchers from the Indian Institute of Technology, Guwahati, have developed a technology to generate green energy during wastewater treatment. The new technology offers the dual benefit of bioelectricity generation and wastewater treatment. Developed under a program supported by the Department of Science and Technology, Government of India, this innovation can be helpful in solving the problem of water and power management in India, which has a rapidly growing urban population. Here is a Science Monitor report.
A growing population and rapid urbanization bring the twin problem of an exponential increase in energy demand and the need for better wastewater management. To add to this, there is the increased environmental impact too. Keeping this in mind, several measures are being undertaken, including the development of renewable and sustainable energy generation technologies. Along with several renewable energy sources, the sustainable energy sources also include the blue energy sources of energy harvesting. These sources of energy generation have no negative impact on the environment, and energy is generated from various wastes such as sewage waste and food industries wastes. In a big step towards using blue energy sources, researchers from the Department of Chemical Engineering, Indian Institute of Technology, Guwahati have developed a bioelectrochemical device. This device, called the microbial fuel cell, treats wastewater and produces green energy. Developed under a research program funded by the Department of Science and Technology, Government of India, this technology can provide dual benefits of renewable energy generation and an efficient wastewater management system.
One of my research students, Mr. Mukesh Sharma and myself, jointly work with some PhD projects. This is a generation of energy from wastewaters. So this energy is a blue energy, and we have developed that bioelectrochemical devices. Basically, this is a microbial fuel cell, and that can generate green energy by treating wastewater. This is simultaneous wastewater treatment as well as generation of energy.
The microbial fuel cell developed by Indian researchers is a bioelectrochemical reactor system that uses electrons liberated during the biochemical oxidation of organic substrates induced by microbes, that is anaerobic bacteria that live in the absence of oxygen, to convert them into electrical energy. The device consists of an anaerobic biotic anode chamber, an aerobic biotic or abiotic cathode chamber, and a separator such as proton exchange membrane. The focus of the present research was on the development of cost-effective and high-performance membranes for application in MFCs. For this, the researchers used sugarcane waste to reduce the cost of proton exchange membranes.
Specialized membrane which is designed by our team is placed over here. What is the property of the membrane is it preferentially allows protons to pass through the anode to the cathode. What happens? The protons which are produced by anaerobic oxidation of the organic matter, it passes from anode to cathode via this membrane, and the electrons they flow through the external circuit. As we can see clearly, the current is being produced.
In the anodic chamber, active biocatalysts oxidize the organic matter present in the wastewater to produce electrons and protons. Protons are brought into the cathodic orbital through the proton exchange membrane, while an electrical circuit is made by conducting electrons through an external circuit. At the cathode, these electrons and protons react in the presence of oxygen, which turns into water. Following this success, work is on to develop a pilot plant on a large scale. This technology development has provided an excellent sustainable energy source along with a more efficient wastewater treatment system. It can be utilized in municipalities and other public spaces.
We, the entire Dibru College family, congratulate Sri Mukesh Sharma sir on his achievement. Thank you.
The device consists of an anaerobic biotech. Good morning everyone, respected principal sir, teachers and my dear students. I am very thankful to sir for inviting me here and to present my work, especially thankful to Jan sir, and I have been so much connected to this place, Dibrugarh. I was here in 2012 and the 1 year that I spent that was spectacular. And in fact, I'm so much happy to be here and present my work over here. Okay, so today I'll be presenting my work, the topic of which will be towards sustainable energy generation and wastewater treatment using bioelectrochemical process. The progress and prospects I'll be discussing on. So this is the outline of my presentation. What we can see here, the prime objective of my research was to use the waste and waste and waste. The idea was, as we can see in this, this is the organic matter. So the idea was to use the organic matter to prepare a specialized membrane. We call it proton exchange membrane. And the property of the membrane, I'll discuss this later in detail, but still I'll tell you, it preferentially allows protons, only exclusively protons, to pass from one side of the reactor to the other side of the reactor. And not just that, I have used the wastewater. Here I have focused on using the municipal wastewater because of the time constraints that we have during the PhD period. So I've used the municipal wastewater, I have treated it, and parallelly I have generated green energy. But when we say green energy, we mean it is having absolutely, as for now, the research says there is no harm to the environment at all. So let us start with the introduction. Now you will be surprised to know that the current scenario, the sanitation scenario in India is, for the rural India, still 70% lack the latrine facilities, and 19% of which are practicing defecation outside, open. Now, the urban area, 67% lack latrine facilities and 12% practice defecation outside. Looks very bad, right? Now, we have an example. A toilet, recently the government has been promoting, okay, you please do it in a proper way and make things more organized. So they are providing funds and all those things. But we are Indians, right? will not improve so easily. So if we can see here, Dr. Suresh Kumar Rohila, he just was no, he was doing one project, and there he visited a number of projects which Indian government was funded, and then see, this is the situation. The funds are there, everything is there, but no, this is for a different person, and the defecation will be in the open only. Anyway, so what is the harmful effects of these things? So first thing is, it poses health hazards. Definitely, waterborne disease will be there. It raises environmental concerns, and then it leads to water contamination. Now, to to cure this, I'll say cure because I'll tell you in details. I design, it's not design, it is a development, basically a setup which we say microbial fuel cell. So the basics of this is, we have an oxygen poor, we say anaerobic, this anode, and then this is the electrode, this is the cathode, which is rich with oxygen, right? And what happens here, we have this membrane, proton exchange membrane, and whatever the sewage, or what you can say the municipal waste, whatever you want to say, it is now given to this chamber. It decomposes here. The proton exchange membrane, it takes the protons, passes from anode to the cathode, and then the electrons move through the external circuit, and then it is energy is generated. Now with this basics, the first and the foremost key player of this complete process is the bacteria. So what happens basically, I'll talk about here the municipal wastewater to limit the the spreadings. So we have this Mr. Clean, we say Savanilla. I'm not from the biology background, but tell you what, this is the most beautiful thing. The organic, no, the bacteria itself decomposes, and we know one very basic thing: nature has everything, right? We only should know how to take it. So this bacteria, what it does, it decomposes, and during so it generates electricity. Now how does it do? The electron transfer mechanism, there are three ways of transferring electrons. First is direct transfer, second electron shuttling, and third is the nanowire. Just a second. Yeah. I hope everyone can see. No problem. So this bacteria, what happens? This bacteria itself, it sticks to this anode, and it forms a biofilm, right? And then when it starts decomposing, then it while decomposing anaerobically, it releases electrons, protons, carbon dioxide, and several other resources which we will not be talking here. Now this is how the electron transfer mechanism takes place. The substrate, when it oxidizes anaerobically, then H plus is released, electrons, and the CO2, carbon dioxide gas. Now the electrons, they pass through this anode chamber, whereas the proton, they start and they get induced by the proton exchange membrane to follow the hopping mechanism. Now this is the hopping mechanism. To understand this hopping mechanism, let me tell you, it's very simple. What happens? We have a proton, then say this is the SO3 minus. What happens? H is also attached to it. What does proton exchange membrane does? It takes the H from here, as we can see, it takes the H from here, then passes it to the next. Then again it follows and follows and follows until the proton reaches the proton exchange membrane, via which it reaches to the cathode chamber. Now this is an animated image of this process. As we can see, the influent, the wastewater. Now this is, I'm showing a cyclic process. There are two types of assemblies that we follow. First is the batch mode setup, and second is the cyclic process. Here I'm explaining the cyclic process so that you understand it properly, what we actually want to do. When influent is given to this anodic chamber, the level of this rises, and then the excess of which passes and then it circulates like this. Now this complete setup, including this, is completely packed. No air is being allowed to pass through it. Now what happens? That cathode chamber is open. There are several assemblies including air cathode, then we have water cathode. I'll be explaining later. So here lots of oxygen is there. What happens? This H plus ions, as we can see, which are produced, they pass through from anode to cathode via this proton exchange membrane, and this electron, as we can see, they pass through the external circuit, and hence we get this electricity. So now there are wide range of applications for this process, including streams, then municipal, this is what I have done, then wastewater, then farmland, Mars, and undersea. Now when we talk about types of MFCs, see, our motive is to provide oxygen to the cathode. Depending on the application, we do it in a different way. For example, for double chambered, we can see air cathode MFC. We can also use here single chambered air cathode MFC. There is no big deal. Why? Because our motive is to provide oxygen or air to this cathode. So what happens here? In the other, we can see single chambered MFC. Here there is no extra chamber. So this is what I'm going to display it here, and this is the other type of MFC without proton exchange membrane. So it's not like proton exchange membrane is essential, it is required, but we can also do it without proton exchange membrane. Anyway, so there are different types of MFCs, including multi-electrode MFC, upflow MFC, downflow, vertical flow, horizontal, mediator, pure culture, and stacked MFCs. Each kind of assembly is having some drawbacks and some advantage. So this multi-electrode MFC is simple to design, and it is popular for scale-up MFCs and produces enhanced power density, power recovery, same thing. Now pure culture, in case of pure culture, the problem is maintaining the pure culture in field scale is very difficult. Sorry. In stacked MFCs, managing voltage reversal is the major challenge. So let us move to the separator, that is the membrane. Now what is the main purpose of the membrane in microbial fuel cell? As we can see, the anaerobicity in the anodic chamber is to be maintained, and this is the prime purpose of the proton exchange membrane. The motive is to keep that anodic chamber free of air. Now the second thing is it has to restrict the mixing of the anaerobic anolyte and aerobic catholyte. Finally, the important purpose, it is for efficient transport of protons and restricting oxygen diffusion from cathode to anode. There are range of membranes available commercially, but the problem is the membranes like Nafine 117, they cost around $1,000 per meter square. Now here is what I decided to focus on my research. Now if you are planning to make something more fruitful, you have to make it more viable. आपको उसको सस्ता बनाना पड़ेगा ताकि मतलब इजीली उसको इकोनॉमिक पर्पस से किसी भी सेटअप को देखना बहुत जरूरी है, सर जानते हैं. सो ये नफाइन 117 का मैंने सोचा एक रिप्लेसमेंट बनाते हैं, एक ऐसा मेम्ब्रेन बनाते हैं. वी शुड I plan to design a membrane which performs equally to the Nafine 117 and is much cheaper to the Nafine 117 membrane. So I designed a proton exchange membrane. Let us know what is the basic of the proton exchange membrane. A proton exchange membrane isolates the anode and cathode to avoid direct contact between them. Then it incorporates several proton conductive functional groups, right? And performs the essential function of conducting protons and rejecting the electron simultaneously. Now, the qualities of good proton exchange membrane includes high proton conductivity, sufficient mechanical strength. It should be able to resist chemicals and higher temperature and different harsh condition, right? And it should be low cost, it should be thin film formality, and then it should be compatible. Now, for making my membranes, there are no two steps. First, as we have discussed, we need functional group. Only then we can attract protons, only then we can repel electrons. To make this possible, I have made modifiers. Two modifiers I prepared. First, cellulose acetate, second, graphene oxide. So as to make my membrane cheaper as possible, I made cellulose acetate from the waste. जब हम अभी गर्मी का मौसम है, जब हम जाते हैं कॉटन कॉलेज के सामने में जाता था, तो I saw that वो गन्ने का रस देता है and then he used to throw that. तो मैंने सोचा okay, let us think about something. I went through the literature and then I found I can make cellulose acetate from it. So I made cellulose acetate from the sugarcane bagasse. Then I made graphene oxide using TOS method. I have mentioned here TOS method, but to be honest, it is still under review. TOS method, although it consumes very less sulfuric acid as compared to the modified Hammers method, I have developed this method further to reduce the cost and to reduce the extent of the exothermic reaction taking place. Because whenever we prepare graphene oxide, we use huge amount of sulfuric acid. So this is how I have prepared cellulose acetate. I'll just go in brief. Initially, the acid treatment was done, followed by the alkaline treatment of this sugarcane bagasse. Then bleaching was done, and lastly, the glacial acid was used to treat it further to form it to the cellulose acetate nanofiber. You can see this is the final product. It works better than the cellulose acetate. Now, this prepared cellulose acetate was characterized using FTIR spectrum, then FESM, Raman shift, and XRD analysis. Here I have compared the commercially available cellulose acetate and the prepared cellulose acetate, and we can see clearly the peaks are more prominent in my case. That means the nanofiber, the cellulose acetate nanofiber that I prepared, it is more effective than the commercially available cellulose acetate. Now, the second modifier, that was graphene oxide. I have mentioned here a method, modified Hammers method, because my method is still under review, so I haven't prepared it here. Mentioned it here. So this method also can be used to prepare graphene oxide. And initially, what we do, we take 2 grams of synthetic graphite and 1 gram of NaNO3. Then we add H2SO4. We can see here 50 ml of 98% concentrated H2SO4 slowly, and then we stir again continuously for 15 minutes. Then 6 grams of potassium permanganate is added. Further, the temperature is maintained to 20 degrees Celsius. Then the mixture is removed from the water ice bath and the temperature is allowed to rise to 35 degrees Celsius. Maintaining temperature here is the key. Only then proper exfoliation of this synthetic graphite to graphene oxide will take place. Now, this mixture is turned to dark brown. Then the process is followed, cleaning process is followed, and then finally we get this graphene oxide. It is like a dark coffee brown kind of a fine powder. Anyway, so the characterization of this graphene oxide is done using XRD and FTR. This is 2 that I have shown here. There are lots of, but just to make it confirm that graphene oxide is formed, this is the main thing, including this Raman, that is also done. So graphene oxide is prepared using this process, and then the membranes that are being prepared here, they are prepared using phase inversion process. What we do basically, we use polymer that we have prepared, that is cellulose acetate. Then we use a base polymer which is comparatively hydrophobic because we all know that cellulose acid का इतना strength नहीं होता है. So to make it to make the membrane more stronger, I have used here a base polymer which is hydrophobic and better in strength, that is PVDF. So the phase inversion process has been followed. Now here wide range of membranes I have prepared. I'll just show 2-3 best of my membranes. Cellulose acid based graphene oxide dope proton exchange membrane. This membrane has no other polymer. Just to initially I want to check that what is the strength of this membrane, so that I can modify it accordingly for strength and other things. So what I observed, that prepared proton exchange membrane has proton conductivity of 0.273 Siemens per centimeter and ion exchange capacity of 1.08 millimole per gram, and this is at room temperature 30 degrees Celsius. Now the developed membrane was 101 USD per square meter, which is very cheap. But yes, we cannot directly compare to the Nafine 117 membrane in terms of mechanical strength and resistant to the chemicals. So what I did, I added PVDF to it. पहले मैंने प्योर CA का membrane बनाया, फिर उसका strength बढ़ाना है. So I added PVDF in small amount, keeping in mind the cost should not go very high because PVDF is a little costlier material. So this membrane was prepared doping it with reduced graphene oxide. ग्राफीन ऑक्साइड को भी रिड्यूस किया ताकि जब मैं सल्फोनेशन करूं तो इतना एक्सफोलिएट हो जाए कि उसमें जो ये सल्फर ऑक्साइड के जो आयंस हैं वो प्रॉपर्ली अटैच हो जाए, वहां पे सल्फेट आयंस अच्छे से अटैच हो जाए. देन द प्रोटॉन कंडक्टिविटी ऑफ 0.4 सीमेंस per centimeter was achieved and the cost, as you can see, was only 434 USD per meter square. From 1000 of Nafine 117, I took it to 434 USD per meter square. The next membrane was made up of PSF based. Initially I used PVDF, here I use PSF. Again GO and graphene oxide and CA was impregnated. Then proton conductivity of 0.31 Siemens per centimeter was there or was obtained, and you can see here the price has reduced further, only 262 USD per square meter, which is very good because this is the proton conductivity of Nafine 117 membrane is actually lower than this. Sorry. Now this is the membrane that I made using sugarcane bagasse. So here we can see the proton conductivity increased. Now this is 0.42 and the cost is 475. Here I found that this membrane can, yes, it can be directly compared to the Nafine 117 membranes, and this is what the patent we are looking for. Now all these membranes was used for studying the microbial fuel cell operation. Now this sorry, so this was telecasted in NERC, Northeast Research Conclave, one of the biggest conclaves that we have in Assam, Northeast completely. Anyway, so let's move further. The prepared proton exchange membranes when used for the microbial fuel cell using municipal wastewater were not just cost effective, but they were also not just very good, I will say. You can just compare Nafine 117 using platinum as a catalyst could only produce 47.6 milliwatt per meter square of the power density, whereas my membrane produced 150.22 milliwatt per meter square power density, and the cost of this was 472 USD per meter square, which is more than half cheaper. And all the membranes that I have prepared I cannot compare this one. Sorry, I cannot compare this one, but surely all other membranes can be compared. So what happens? Every process has some advantage and disadvantage. But initially I discussed that my process has none. It is only having advantages, no disadvantages. What happens? Post process, whatever residue is left, even that is a gold. How? Let's see. The sludge that is managed, important factor behind, okay, sorry. The sludge that is managed in this generation, in this power generation process in such a way that it is either used as a manure, right, or the amount of sludge is so much small that we can effectively neglect it initially, and then after some time the bacteria itself decomposes it further and further and further, and even this is dissolved and then it is gone. So now what is the future? As I have said, that this process is not just for power generation. This process is not just for wastewater treatment. It is about extracting so many things from the waste. When we talk about waste, we do not mean only the municipal waste. We mean waste from several industrial sources too. Now, this could be petroleum, or then it can be used, say I'll tell you, leather industry, sugar industry, right, and various other industries, including, in fact I would tell you, the research is going on to treat industrial wastewater from Tata Steel. We have obtained that wastewater from Tata Steel and we are working on that, and till now whatever results that I have seen with my setup, it is awesome. But I cannot discuss that in much detail over here because that is an unpublished work, in fact. But anyway, so many things can be generated at the cathode output, as the cathode output, including electricity, which I have done, hydrogen, methane, hydrogen peroxide, valuable organics, including NaOH, HCl, struvite, algae biomass, and clean water. Now, along with this, desalination can also be done. When we say desalination, then we mean such a big thing. Why? As we all know that there are two major crises for the future: energy and water. If we could successfully desalinate the seawater, the ocean water, let's not talk about ocean, that will be a very big space. If we can desalinate seawater, even that could be a very big thing. Even if we can make that water in such a way we can consume it, that is a very big thing, because we all know the consumable water that we have in the earth is very small. So using this process, desalination can also be done. That is why I have placed here X, because here I have used microbial fuel cell. It can also be microbial electrolysis cell. It can also be microbial desalination cell. So what is the main thing? Main thing is I can use micro for so many things. There is a range of application that can be done using this. Not just this, anode input. Just see this. There is a range of inputs that we can provide to it, including ammonia, ferrous iron, sulfide, biofuel, root exudates, then residues, and petrol, sediment, wastewater, and so many other which is still under study. Show a setup which has two chambers, anode and cathode. But as I have already discussed here, we can have a different mode of assembly. So the simplest mode that I will show you here is this setup. You could see here, this is just the anode. इसमें cathode भी नहीं लगाया. क्या किया है मैंने, जो ह्यूमन वेस्ट होता है, म्युनिसिपल वेस्ट वाटर जो IIT गुवाहाटी का है, उसको मैंने लिया, सिंपली उसको इनोकुलेट किया, ताकि जो अननेसेसरी बैक्टीरिया है जो मुझे नहीं चाहिए. प्लीज कीप इन माइंड, व्हेन वी से बैक्टीरिया, then keep it on, when we say bacteria, then there is a range of bacteria for different purpose. For wastewater treatment we have different bacteria, for power generation we have different bacteria, और वेस्ट वाटर ट्रीटमेंट में भी कई बार ऐसा होता है अलग अलग मेटल्स को हटाने के लिए अलग अलग बैक्टीरिया मिलता है. सो माय ऑब्जेक्टिव वाज, आई वांटेड ओनली मिस्टर क्लीन एंड जियोबैक्टर ओवर हियर, तो बाकी सबको मैंने इनोकुलेशन करके सप्रेस कर दिया. राइट, अब देखिए इसमें हमारे पास एनोड है जिसको परफेक्टली सील किया हुआ है ताकि कोई भी इसमें हवा ना जाए, और देखिए इधर कैथोड. अब बोलेंगे ये yeah, I'll tell you so that let me finish it. So here we can see this is cathode. Where is cathode? अरे हवा में, पूरा हवा ही कैथोड है भाई. राइट, दिस इज द इलेक्ट्रोड. ये भी सबसे सस्ता इलेक्ट्रोड में से एक है. दिस इज कार्बन क्लॉथ. अगर इसकी परफॉर्मेंस और बढ़ानी है, वी कैन अटैच प्लैटिनम टू इट. एज वी कैन सी, वहां पे मैंने दिखाया था नफाइन 117 यूज किया था लेकिन प्लैटिनम लगाया था. यहां मैंने अपने मेम्ब्रेन लगाए हैं, कोई प्लैटिनम की जरूरत नहीं है क्योंकि प्लैटिनम बहुत कॉस्टली है. अभी भी स्टिल रिसर्च इज गोइंग ऑन टू मेक अ ग्रीनर अप्रोच टू एक्सट्रैक्ट, और यू कैन से प्लैटिनम बनाने के लिए ग्रीनर अप्रोच चल रहा है जो कॉस्ट इफेक्टिव हो. बट स्टिल इट इज having a lot of time, I believe. Apart from this platinum, studies are also going for MOF. आप लोग केमिस्ट्री वाले कितने हैं I don't know, but MOF is a metal organic framework, right sir? उसकी जो सरफेस एरिया होती है वो इतनी ज्यादा होती है. आप एक चीज देखिए, यहाँ पे जब मैंने डिस्कस किया था SO3 - H+ का, क्या होता है कि SO3 और H, इन दोनों का जो डिस्टेंस होता है ओनली 1 एंगस्ट्रॉम होता है, 1 एंगस्ट्रॉम. और कई ऐसे एटम होते हैं जिनका आप वो देखिए क्या बोलते हैं डायमीटर, वही उससे कम होता है. वही मतलब क्या होता है, उसके बीच में जो बॉन्ड है इतना वीक होता है, इट कैन इजीली बी ब्रोकन. और प्रोटॉन एक्सचेंज मेम्ब्रेन जितना एफिशिएंट रहेगा, बॉन्ड उतनी जल्दी टूटेगा. और H+ को जितना आसानी से प्रोटॉन एक्सचेंज मेम्ब्रेन एनोड से कैथोड में लेके जाएगा, इलेक्ट्रॉन उतना ही रैपिडली बढ़ेगा. दिस इज व्हाट द मोटिव इज. सो एज सर आस्क्ड हियर, सर योर क्वेश्चन अगेन, आई जस्ट फॉरगॉट. Yeah, now I remember, sir. Here, sir, since my background is not biology, right? So I just followed several literatures. I focused on only a single category of bacteria, right? Geobacter primarily. Then Mr. Clean. So मैंने ये एक process जब देखा, several researchers followed a single process, right? To suppress all other microorganisms, all other bacteria, they inoculated it using chloroform. So what basically we do, we take, for example, a kilogram of this, I have wastewater in a big jar. I put 3 to 4, I just boil it for, boil means I heat it for 5 to 10 degrees Celsius above the room temperature. And then I pour 2-3 drops of chloroform and I keep it for overnight minimum. And then that's done. Then we can simply take that from syringe and then that's it. So yes, yes. Main thing is, sir, the more we complicate the process, right? The more we require, no, ये technology चाहिए, वो चाहिए, नहीं, वो सब नहीं चाहिए. Let us make it simple. Only then it will be approachable. if we want to develop this technology, we should work on the catalyst, we should work on the separators, membrane, because वो एक मैंने जो माइक्रोवेव फ्यूल सेल दिखाया था which was without membrane, एक्चुअली काम तो उससे भी चल सकता है लेकिन वो एफिशिएंसी नहीं आएगी in terms of power density, in terms of wastewater treatment. So we need membranes, we need catalyst, and that is the only two things that we are at this stage focusing on.
Density will also impact the flow.
Definitely, sir. Definitely.
On the way, you can just pump up the bacterial culture and then decomposition will move and then
Yeah, definitely.
the permeability of the membrane.
It is one of the factors, but sir, to be honest, what happens? We are following a cyclic process, right? So the bacteria that we have cultured in anodic chamber, एक टाइम आता है जब उसकी efficiency घट जाती है, right? So what? Because wastewater is treated, तो जब पानी साफ हो गया तो उसकी जो जब तक गंदा है तब तक अच्छा है हमारे लिए, जब साफ हो गया that is a waste, बेकार है अभी, तो we flush it out, फिर से डालो भाई, क्या प्रॉब्लम है? So aim is, you could see आजकल बिजली का दाम कितना ज्यादा हो गया है. So my aim is to make this technology to such a stage, सबके घर में, चाहे वो गरीब से गरीब आदमी हो, उसके घर में एक मतलब पॉटी करने वाला वो सीवेज टैंक होता है. तो माय पर्पस इज कि कैसे भी करके इस टेक्नोलॉजी को इतना डेवलप करना है कि वो जो ही इज यूजिंग दैट टॉयलेट, then simultaneously he will generate energy so that at least 2 to 3 LED bulbs can be lighted, and then at least 1 to 2 DC motor fan can work, and that is the biggest achievement we can have. There are lots of people, the majority of the population I would say, अभी भी जो बिजली बिल आता है तो उनकी सैलरी का एक पोर्शन रहता है वो, right? So my purpose is to solve that problem. Using that motive, I am going through this research.
So that flow you had made for the down to up.
Yes, sir.
If I change the geometry, will efficiency change?
Yes, sir. It would. I'll tell us a simple thing. We follow gravity, right, sir? So whenever this waste will come, it's not a complete liquid, right? It is a concentrated sludge. In any case, it will settle down, right, sir? So when it will settle down, only then it will form biofilm. It is important. I'll go into more insight. It is very important. I'll show you one thing, sir. So this is the membrane, sir, right? This is the active layer and this is the passive layer. So we place this active layer this side towards the anode side, right? क्योंकि ये जरूरी है कि एनोड में जो भी पानी है या वेस्ट वाटर कुछ भी है वो कैथोड में नहीं जाना है. This is very important. We want to restrict the flow of any kind of transport of anything from anode to cathode except protons, right? So we will be placing here the active side and here the passive side. So what happens, जब सर जैसे सर ने बोला हम जानबूझ के जो इन्फ्लुएंट है वो नीचे से दे रहे हैं ताकि हम चाहते हैं ये तो this setup is small, but when we make a larger setup we try to expose the maximum membrane to the anode, right? So we want कि जो भी sludge है, concentrated sludge है, we want that to settle down for some time, get decomposed, form a biofilm over here. Only then Mr. Clean and Geobacter will function properly, right? जैसे जैसे देखिए यहाँ पे बायोफिल्म फॉर्म होगी एंड देन जब ट्रीट होते जाएगा तो बिकॉज इट इज साइक्लिक प्रोसेस इट विल अगेन बाहर निकल जाएगा, फिर आएगा वो गंदा पानी, फिर यहाँ पे बायोफिल्म फॉर्म, फिर ट्रीट होगा, फिर इट विल फॉलो लाइक दिस. आप जो कह रहे हैं सर ऊपर से ऐसे ले आओ, आई वुड से सर बिकॉज ग्रेविटी का काम तो होगा ही, अगर हम ऐसे घुमाएंगे उसको ऊपर से नीचे ले आएंगे तो जो बैक्टीरिया वाला जो पोर्शन है जो हम चाहते हैं कि थोड़ा देर तक रहे इसमें वो नहीं रहेगा, राइट? हम चाहते हैं कि वो थोड़ा सेटल डाउन रहे, तब तो यहाँ फिल्म फॉर्म होगा. फॉर दैट रीजन we are making it like this, not like this, right, sir?
One of the other factors to increase the power density. So with the power density that even talked about,
Yeah.
because the power density want to deliver in the output, right?
Yeah.
So what are the factors that like what makes it different from the membrane diaphragm?
Nafine 117. Sir,
Power density high, too high.
so the first thing is, कोई भी membrane का sir, proton exchange membrane I'm talking about, the initially during my presentation I told you, for a proton exchange membrane to be efficient and effective, it is important to have good strength in terms of mechanical, chemical, temperature, so many things. Then it should have high proton conductivity, then it should have high ion exchange capacity, it should have हाई वाटर अपटेक. अब देखिए ये भी कॉन्फ्लिक्टिंग चीज है, आप बोलेंगे कि उसका वाटर अपटेक भी हाई है फिर भी आप पानी को उधर पास नहीं होने दे रहे हैं. यही तो खासियत है मेम्ब्रेन की, नहीं तो हम सिंपल फॉल्ट सोर्स मेम्ब्रेन लेके नहीं लगा देते. इट इज हाइड्रोफिलिक, या क्योंकि अगर हम, दैट इज द मेन थिंग सर. एक्चुअली 100% तो रिजेक्ट नहीं करता है पानी को, क्योंकि कोई भी प्रोसेस 100% एफिशिएंट तो नहीं हो सकता है. आई एग्री विद दिस, लेकिन नेग्लिजिबल है, गीला करता है. इसीलिए मैं आपको बताया ना सर, आई एम यूजिंग मॉडिफायर हियर, प्लस आई एम टॉकिंग अबाउट, या that is the reason. There are two reasons for using hydrophobic polymer: A, strength; B, I want a little hydrophobicity also. Secondly, if we use जैसे पॉलीसल्फोन यूज करते हैं, ऑलरेडी उसमें सल्फोनेट ग्रुप्स हैं, तो ऑलरेडी उसमें चार्ज आयंस हैं, राइट. पर्पस इज, जो प्रोटॉन एक्सचेंज मेम्ब्रेन है उसके सरफेस में आपने जितने भी केमिस्ट्री वाले हैं या जो फिजिक्स वाले भी हैं, आप ये सोचिए कि मैं क्यों ग्राफीन ऑक्साइड यूज किया. द मोटिव वाज, मुझे मेम्ब्रेन के सरफेस में नेगेटिव आयन चिपकाने थे. एज सिंपल एज दैट. जब नेगेटिव आयन चिपकाऊंगा तब तो वो पॉजिटिव को खींचेगा, है कि नहीं. जब नेगेटिव आयंस उसमें रहेंगे तब तो वो पॉजिटिव को खींचेंगे, और पॉजिटिव को खींच के वो, जैसे ये मैं मेम्ब्रेन हूं, पॉजिटिव इधर है, लो भैया, पॉजिटिव इधर है, लो. दिस इज व्हाट इज डूइंग. अगर नेगेटिव आ गया ना समझाओ. So that is the main purpose and that is why I have used graphene oxide. और आप लोग एक जान के हैरान हो जाएंगे जितने केमिस्ट्री वाले हैं कि जो सेलोज एस्टेट है उसमें नेचुरली, जो सेलोज एस्टेट होता है उसमें नेचुरली नेगेटिव आयंस अटैच्ड होते हैं. आप उसके स्ट्रक्चर को अगर फॉलो करेंगे तो सर COOH होता है ना एसीटेट का, तो भी तो नेगेटिव है. तो वो खुद भी, इसीलिए वो मैंने वहां यूज किया. एवरीथिंग हैज बीन यूज्ड हियर विद अ पर्पस. कोई ऐसे फ्री में आके नहीं बैठ गया वहां पे, किसी मोटिव से वहां बैठा है. सेलोज एस्टेट का दो काम है यहाँ पे. एक, आपकी मेम्ब्रेन की हाइड्रोफिलिसिटी बढ़ाता है. बी, वो प्रोटॉन एक्सचेंज कैपेसिटी और प्रोटॉन कंडक्टिविटी को भी बढ़ाता है, राइट. So this is the main thing. Any other question? I am expecting question from the student side. हाँ.
So you have talked about generation of electricity.
हाँ.
So can you tell about the amount of electricity that is generated in terms of unit and the amount of, suppose you are putting 1 liter of wastewater.
Okay.
So how much it is generated? And how you can make it more efficient?
Very good.
Is your membrane efficient? That 117 that you have talked about, that will be more efficient to generate electricity.
Okay.
Because the main final the product that we want, we need electricity.
Definitely.
In every home of India.
Yeah, definitely.
How it will be efficient for us?
Well, I'll tell you. See, here, if we talk about, now fine, say this setup, this is a small setup, right? ऐसे ही लेके इसको हम LED बल्ब में लगा दिया, जला दिया, वो नहीं होगा, right? वो why? पहली बात, we will need to scale up the setup, right? When we need to scale up the setup and make it to the commercial level you're talking about, for that we need, जो membranes हैं उनकी proper assembly, design and so many things needs to be done. फिर वो डायरेक्टली हम उसको यूज नहीं कर सकते हैं, उससे भी जो पावर जो होगा जनरेट होगा, वी नीड टू स्टोर इट प्रॉपरली. जैसे इस केस में मैंने क्या किया है कि कैपेसिटर में पहले जो सुपर कैपेसिटर होता है उसमें इसको स्टोर किया, ठीक है. और सुपर कैपेसिटर से हम डायरेक्ट एलईडी में नहीं देंगे. सुपर कैपेसिटर क्या करता है, जितनी एनर्जी उसके पास है, तुमको एनर्जी चाहिए, ये लो पूरा दे दिया. आई डोंट वांट इन दैट वे. हमें क्या करना है, सुपर कैपेसिटर को चार्ज किया और सुपर कैपेसिटर से हम बैटरी को चार्ज करेंगे, सो दैट वी कैन कंट्रोल द फ्लो ऑफ करंट, राइट. ओनली देन वी कैन यूज इट फॉर लाइटिंग बल्ब. इससे राइस बल्ब जो आता है एलईडी वाला, वो मैंने जलाया है वो यूज करके. नफाइन 117 से भी अभी तक लोगों ने जो भी काम किया है स्केलिंग का, उसका आई कैन नॉट टेल यू द एग्जैक्ट डेटा, बट जो हमारा रिजल्ट है इस मेम्ब्रेन का वो उससे अच्छा है, राइट. वो उससे अच्छा है. अब कितना है, वेरी सून विल आई विल हैव दिस, यू कैन फॉलो मी and then you can see the results in this papers itself, right. But that is the last thing that we have to do, क्योंकि उसके बाद तो we can process it for commercial assembly, but before that हमें अभी बहुत कुछ काम करना है. क्योंकि बेटा देख, थिंग इज, जब इनिशियली सोलर पावर जब जनरेशन शुरू हुआ था, नो वन बिलीव कि अरे उससे भी पावर ले सकते हैं क्या. तो ये भी उसी स्टेज पे है. अभी कोई अरे कैसे तुम 40 से एनर्जी बना सकते हो, बट आई हैव शोन इट दैट वी कैन डू इट. तो अगर उससे कर, सूरज से भी कर सकते हैं तो इससे भी कर सकते हैं, कोई प्रॉब्लम नहीं है. या इट विल टेक सम टाइम, मोर इन डेप्थ रिसर्च विल बी देयर, बट आई होप विद इन 5 to 6 years दिस विल बी देयर, राइट. थैंक यू. एनी मोर क्वेश्चंस?
Is IIT
Yes, sir.
collaborating with any industry for development of?
Sir, we are actually we are having projects in collaboration with Taiwan. Yeah, Indo-Taiwan project. And recently, my professor, Professor Purkit, he has now been assigned as secretary or some supreme post of this Jal Jeevan mission for this Assam region. So we are also with that project that we are working on this technology. So many projects are going for this, but at this stage, I think it will still take 1 or 2 good years, because the main thing is, sir, resources. You see this setup, sir. ये कैसे कैसे वो जो जुगाड़ू कमलेश वाला है ना, वही पूरा काम चल रहा है. In fact, I will tell you the main thing. Before starting this, I didn't. Please don't take this in other sense. मैंने अपने प्रोफेसर को ये भी नहीं बताया कि आई एम गोइंग टू डू दिस, अदरवाइज ही विल से दैट नो we don't have expertise people in our lab and we don't have setup, we don't have instrument. Once I did this, in 3 months I got good results. Then I shown to sir, this is the result. Okay, very good, write it in the form of paper. So unless you have the results, your research is useless. And if you can't put your science to application, then your every study is just पेपर में लिख देने से, रिसर्च पेपर पब्लिश करने से साइंस नहीं होता है. दिस इज व्हाट आई बिलीव. मेकिंग इट इनटू अ प्रैक्टिकल फॉर्म इज द एक्चुअल साइंस. थैंक यू. थैंक यू सर.
Thank you, everyone. So now we shall wind up the program. I'd like to thank our principal, sir, for allowing us to organize this program. Thank you very much, sir. I thank Mukesh Sharma for his beautiful presentation. He took the pain to come up to Dibrugarh from Guwahati during the rainy season. That was very tough. So, and we have good wishes for his future endeavors. Thank you, Mukesh. I thank all our esteemed faculty members for being present here. And I also thank all the students for listening to the presentation very carefully and also interacting with the resource person. I like to thank our non-teaching staff, especially Deepak, Vikas and Amit, for
arranging the setup. Thank you once again.